All right, hello everyone. This is the last video I think I'm going to do for my spring break series. Spring break is actually over now, but yes, this is the last of the videos that I was planning to do to review the exam 2 content over spring break. This is chapter 5, dominance, the different kinds of dominance. So it's very often that we see, and it's pretty intuitive that many organisms' genes are coded for not just by like a single instruction. You know, for example, we're diploid, so almost all of our genes have a second allele that is representing and coding for the expression of that gene. And so, different kinds of alleles have different tendencies. Right? Some alleles have the tendency to overpower other alleles. And this is, like I said, pretty intuitive. And we come across a very familiar situation of complete or conventional dominance. And this is where one kind of allele is dominant over another kind. And so we see at the B locus, for example, in cats, which has to do with the color of their fur, we have a capital B allele and a lowercase b allele. Capital B is dominant to lowercase b, and so we see that in an individual who's homozygous dominant, obviously they'll show the corresponding phenotype. A homozygous recessive individual will show the corresponding phenotype. But a heterozygous individual will show the dominant phenotype because they have a dominant allele and it, dominates the recessive allele. And therefore they express the dominant phenotype. Oops. Where'd that thing go? Why does this always happen? Go away. Goodness gracious, do I really have to do this? It's fine. We just need to be patient. It should automatically hide itself, so I don't know what's going on. Could I please see my thing? Please? Please go away. I do kind of need to see the little the little thing at the bottom so I can tap the different colors. Grr. Hey, taskbar, go away. Ugh. Actually, I should have brought that up in the first place. Sorry about that. Okay, so now the taskbar goes away. Ugh, typical. Let's find our way. We're never lost as long as we're patient. Okay, that's better. Now could the taskbar come back for just a little bit? Hey, there we go. Hey, we did it. Okay. So this is what we were going for. Ding dong, ding dong. So this dominant B allele, if it is present in this diploid individual at the B locus, the dominant, that is very thick, the dominant allele will dominate the phenotype that is expressed by this heterozygous individual. That is, excuse me, that is complete dominance, the very familiar, conventional kind of dominance that we come across a lot. And so it's easy to think, the capital letter will dominate the lowercase letter. It's not always going to be capital and lowercase. For example, you could have, we see it commonly in sex-linked traits that you have maybe a gene that's A, but there is a wild type and a mutant type. Typically, the mutant type is dominant to the wild type. There could be, it's arbitrary. You could have A star versus A sub, B versus, it could be any kind of combination. A alpha versus A gamma, you get the point. But it's just very simple. One allele dominates another. And in individuals with both kinds of allele, the dominant genotype will dominate the phenotype. There's another kind of dominance, though. It's not just the very nice, clear-cut, complete dominance. We can have what is called incomplete dominance. And this is where you see kind of a gradation, an intermediate series of phenotypes based on how many dominant versus, or I shouldn't say dominant, actually, yeah, dominant versus recessive alleles that you have in an organism. So let's look at, once again, these cats. Cats, once again, at the B or the color locus, have either a capital B, which is dominant to a recessive lowercase b, but it's not entirely dominant. Having a single capital B does not show the dominant phenotype. That is, or that corresponds to the dominant capital B allele. Instead, we see an intermediate phenotype where having, all big B's makes you black, having no big B's, instead having little b's, makes you white. And in this example, we can think of any one dominant B allele it codes for like 25% of the possible black pigment to be deposited into the hair of a cat. So we have 25, 50, 75, 100%. We have each of these. Oh, that should not have been the eraser. We have every allele contributing to the deposition of pigment. But in this case, we only have 25, 50, 75%. And so this individual is not entirely black. They are an intermediate between the dominant phenotype, the homozygous dominant phenotype, versus the homozygous recessive phenotype. And as you go along, you see that fewer of the alleles are capital B, fewer are dominant, and therefore there are fewer that are, pushing pigment into the hairs of these cats. Only 25% here and 0 here. And when I think of incomplete dominance, or when I see an intermediate phenotype, I think of incomplete dominance. And so this is a special case. Here in the conventional or complete dominance, we see a 3-to-1 ratio. If we were to do a monohybrid cross, B versus B. Well, actually, no. It would be a homozygotes. So BB and BB, this will create a 3-to-1 phenotypic ratio where 3 of them have or show the dominant phenotype and 1 of them show the recessive phenotype, because we'll have capital B, capital B, capital B, lowercase b, capital B, lowercase b, lowercase b, lowercase b. And so we have a capital B here, and 2 capital B's here. So having the capital B, because capital B dominates lowercase b, that is going to display this black or dominant phenotype. And that is what 3 of these 4 individuals are. And then 1 individual doesn't have the dominant allele. They are homozygous recessive, and so they'll show the recessive phenotype. In this situation, this shows that despite having a 1-to-2-to-1 ratio of genotypes, where 1 is homozygous dominant, 2 are heterozygous, and 1 is homozygous recessive, the phenotype is different than the genotype. The phenotype is that 3 are black and 1 is white. But here in incomplete dominance, because we see a series of intermediate phenotypes that increase or decrease in gradation, depending on the quality or the gene that's actually coding at that locus, whatever it may be. It's not always going to be the deposition of color. It could be like the number of fingers that you have, or the length of your tooth or your claw if you're a crab or something. Use your imagination. But here in incomplete dominance, we see that we have 1, 2, 3, 4, 5 different genotypes, having 4 big B's, 3-to-1, 2-to-2, 1-to-3, and 0-to-4. So we see a 1 to 1 to 1 to 1 to 1 genotypic ratio, but because we see series of intermediate phenotypes, every genotype creates its own unique intermediate phenotype that is somewhere in between the homozygous recessive and homozygous dominant phenotypes. And so we also see a 1 to 1 to 1 to 1 to 1 phenotypic ratio. So with incomplete dominance, the genotypic and phenotypic ratios are identical. And yet, in this instance, think of every capital B allele contributing 25% of the possible melanin into a hair follicle. And, intermediate, incomplete. And there's even another kind. It's my favorite personally, but it's a little bit more contrived, less intuitive than the other kinds, and it is called codominance. And this can be kind of like unfamiliar, uncomforting, or discomforting, excuse me, because usually when we think of dominance, we think, oh, yep, one thing is going to overpower another. And in this case, one thing is going to do something and the other thing isn't going to do something. This capital B is going to attribute 25% of the pigment, and this lowercase b is going to contribute nothing. But in the case of codominance, we have two things, two different alleles that are contributing. So here we have an individual. The B allele is codominant with the O allele in individuals that express it. So here we have a BB individual, black pigment, black pigment. Here we have another homozygous individual that has orange pigment, orange pigment. But when we see a heterozygous individual, one that has two different kinds of alleles, one B, one O, B does not dominate O, and O does not dominate B. Instead, you find them equally expressed. They equally dominate the phenotype of an organism at this specific locus. For example, you don't see an intermediate phenotype that's a really dark orange because you have orange that's being added and then black that's also being added that makes it darker. That's not happening. What we see is everywhere that it's black, black is dominant, but also everywhere that it's orange, orange is dominant. One orange is not dominating, one color isn't dominating another. They're codominant because they coexist on this organism. And this is what we see, for example, in calico cats. They have, is there a white option? Bing. Oops. No, kitty. Hey, go back, please. Thank you. So let's change this. So we have calico cats, and they have black, white, and orange spots all over them. And that's because, should I get into that now? Yeah, let's get into it now. Let's see. That is because of something called dosage compensation. So on the whole, well, actually, I shouldn't get into that. And actually, that's not even the same. Never mind. Oops, I almost messed that one up. So we have individuals that are, pretend there's also the white allele. So codominance is where different alleles coexist in their dominant state on an organism, and both alleles are expressed in their dominant state. There's not an intermediate phenotype, but instead a phenotype that is mottled, or I shouldn't say mottled, that indicates or demands a color. Actually, I'm just gonna stop talking. But anyway, so yeah, these are the three main kinds of dominance that we'll see. There's a fourth kind that we don't have to learn. It's called overdominance. And I don't think we talk about it, so let's ignore it. Excuse me. And a common question type that you'll be asked for codominance is about blood type. Blood type, we talk about it in lab, or you guys see it in lab a lot, where you have the I's, the A, B, and the O, or actually this isn't I-O, it's lowercase I. So you can have an individual that's I-A, I-B, and they will have an A-B blood type. They have two different alleles, I sub A and I sub B, and this creates a codominant phenotype, or a phenotype where A is present on a blood cell and B is also present on a blood cell. They coexist in their dominant forms or states. And you also have this with this, which can be written as this, where you have the lowercase I that corresponds to an O blood type. And so you can have this individual, which will have an A blood type, because technically this is codominance because you have the A allele, where you have the A sugars on your blood cells, but this allele is also coded for. It successfully is expressed on this. It just so happens that this allele is just not coding or not adding any sugar. There's no special sugar on this. So it's codominant, but in a less apparent way. And you can also have the blood type IO, which is also two lowercase i's, you can write it as, and that's having a blood cell with, no sugars, A, B, or otherwise, because these alleles code for no sugar added. So if you see blood type, think codominance. And if you see calicos, think codominance. That's another good way to think of it. Codominance, calicos. And actually, I think I was wrong earlier. I should have believed in myself. This is still codominance. Let me bring out the pen. So, in cats specifically, we can very visibly see an important genetic phenomenon, and that is what we know as the Lyon's hypothesis, or Lyon hypothesis. And there was a scientist, a geneticist, her name was Murray Lyon, and she was doing some genetics work in the 20th century, and she discovered, actually no, there was a man named Murray Barr, he was Canadian, he discovered these dark little spots on microscopic cells. And he's like, oh, I'm going to name these after me. So we have these Barr bodies. For a long time, no one knew what they were. But in the 20th century, a geneticist named Mary Lyon postulated that, I think these are X chromosomes that are inactivated. Whoa. And, I am a human female. Let's pretend that these are human female cells. And so we know that this cell is going to have an XX genotype. Sorry, I write my X's in a strange way. But so we have these XX phenotypes, or excuse me, genotypes, but these aren't both going to be expressed at the same time. You might think, oh, we have two X's. That means we can make twice the stuff. Yay. But no. Let's think if you have a liver cell, and that liver cell is going to be full of lysosomes. They're running around, they're full of enzymes, they're destroying stuff. If you have twice as many now, you might destroy too much stuff, and then your cells don't have what they need. Or you might, I'm sure you get the point. Having twice as much stuff is not as good, because our cells need specific amounts of certain chemicals to function properly. And coordinate in an effective manner with their neighboring cells and neighboring kinds of tissue. So we don't want to have both of these X's active at all times. So we still don't know the mechanism. Geneticists haven't entirely discovered how this happens. We don't have a perfect grip on it, but one X chromosome is going to get wrapped up, condensed into this bar body. And so when that happens, we have an organism or a cell that has just a single X chromosome doing its stuff. And this is called dosage compensation. You know, we don't want to overdose on all of the products that come with both X chromosomes being active. So we're going to reduce the dose that we get or that our cells have access to. So with this dosage compensation and the creation of bar bodies via the Lion hypothesis, we can see things like the coloration of cats. This is a tortoise shell cat. And I guess someone looked at this and said, "Oop, looks like a tortoise." But we see, if we zoom in real nice and close, instead of seeing the, oh, I forgot I have to get rid of that. Please bring it back. Excuse me, sir. Hey, come back here. Nope, stop. Hey, please. Okay, I'm never hiding that thing again, goodness gracious. How do you get it back? Oh my God. Oh, goodness gracious. Please just show me the thing. Ah! Okay. Yes, I like this background a lot, but I would even more like to see the damn thing. Oh my God. Okay, never mind. I forgot I can just do that. So, that cat that we saw, the tortoise shell, is this. What's happening in that cat is the same thing that's happening in this cat, in the calico, except instead of having, large patches of uninterrupted coloration here and here and here, instead, like all of the different, or a lot of the different hairs instead of having large uninterrupted patches of color that are the result of codominance, we have individual cells that deposit pigment into the hair follicles. These individual cells are codominant. Coloration is expressed in a codominant way in these cells. So they aren't large uninterrupted patches of color. They're the individual cells that apply pigment to the hair follicles. They are codominant. And so when we zoom out and see that, and I really wished that it would just show me that. Okay, never mind. So we see this where a hair right here, let me bring out the pen so you can actually see it. Nope, come back. Okay, so we see like the hair here, or the hair here is orange, and then the hair here is black, and then the hair here is orange and black and orange and black, and you get the point. You can see it. And the reason that they have this appearance that's so irregular, the reason that any orange can be next to any black hair, which is next to any black hair, which is next to any orange hair, is because as far as we know, it's random, or I shouldn't say it's random as far as we know. We simply don't know how the cell decides which of the two X chromosomes to deactivate and turn into a bar body. We just don't know that yet. And so we can, for the most part in this class, just assume that it's random. And therefore we have a random distribution of orange hairs versus black hairs. And orange and black alleles are each codominant, or they're codominant with each other. What else did I want to say about that? Oh yeah, I guess it does say here that it's randomly inactivated. But we just say that because we don't entirely know. So now that we have that under our belt, oops, it just refuses to go away. Actually, let's make this guy small. Please be small. Please. Never mind. Okay, it doesn't want to be small. So, but we do want to make this guy big. Oh yeah, this is just another representation of the dominance in case you don't like cats. You have this conventional dominant where in the A allele complex, let's pretend that A sub 1 is dominant to A sub 2. So if we have A sub 1 and A sub 2 in a heterozygous genotype, we're going to display the dominant phenotype, which is the A sub 1, which codes for a red color. And in complete dominance, we see an intermediate phenotype, a gradation as you go from this over here to this side over here. You gradually have fewer and fewer alleles that deposit this nice red pigment. And so you have a lot of pigment here, a little bit less here, even less here, and then you have an almost white flower here. So that can become small again. Small. Go away. Ugh, why does it do this? Okay, so now let's have a little example. Is this recording? Hooray, okay. Let's have a little example. Purple. So, excuse me, in a certain breed of cat, coat color is codominant and sex-linked. One allele produces black fur and the other allele produces orange fur. That sounds familiar. Cats that are heterozygous have tortoise shell colored fur, black and orange hairs. If you cross a tortoise shell colored female with an orange male, various things are going to happen. Let's explore. And it always does this to me. So let's make that go away. So let's reiterate what the question has told us. Oh, goodness, I forgot it's going to do that. Be very obstinate. Okay. Anyway, as I was saying, so we have a tortoise shell colored female and an orange male. So these are sex linked. So we're dealing with X's and Y's. Cats have females that mate with males. And so we have this, the B and O alleles to look out for. And when it says sex linked, let's assume that it's X linked instead of Y linked. So we have this X-linked trait that is fur color. We have the B allele that codes for black fur, O that codes for orange, and these are linked to the X chromosome. So we have our female parent and our male parent. The female is something that I should redraw. The female is X, X, and the male is X, Y. So the female is tortoise shell. How do you get a tortoise shell phenotype? By having, well, firstly, we know that tortoise shell phenotype is the result of codominance between the orange, or between orange and black alleles. And so this female is going to have at least one orange and at least one black allele. We're diploid, so we have two X chromosomes. Let's say that this is going to be XB and this one XO. And this female mates with an orange male. So obviously, since this is X-linked, we assume the Y chromosome is not going to have anything to do with color. The color gene is not present on this Y chromosome. So it's going to be on the X chromosome, the only X chromosome that this male has. So these are our parental phenotypes. And if we cross them, let's see what happens. So we can do a cute little Punnett square. We have XB. I'm gonna redo that. Sorry. XB and XO from the mother, XO and Y from the father. So we have XO, XB, XO, XO, XB from the mother, and then Y from the father, XO from the mother, Y from the father. And so what do we see? This individual is XOXB. Firstly, what is their biological sex? They are female. They have two X chromosomes. Cats are mammals, and mammals, the female is the homogametic species. So we have this female. They have one orange allele, one black allele. And we know from even the mother that this codominant situation is going to end up with a tortoiseshell phenotype. So individual number 1 is a torty female. Individual 2 got the orange allele from the father, or excuse me, from the mother and the father. So they are a female, they're two X chromosomes. So individual number 2 is going to be entirely orange, two orange alleles. So moving on to individual number 3. What is their sex? Male. Exactly. Y chromosome determines them to be a male. And the mother gave their black allele in this case. So this is going to be a male with black fur. And then individual number 4, or 1/4 of the progeny, are possible to have this genotype. So the mother gives the orange allele this time instead of black, and the father contributes the Y chromosome. So we have orange male, okay, so these are the outcomes of this cross. So now we can answer the question, what percent of their offspring will be orange? We can look, we have 1 orange, 2 orange. So 2 out of 4, 50%. I'm not comfortable with how that looks. Let's say 50%. What percentage of their offspring will be black? Only 1 black. Out of 4 is 25%. What percentage of their offspring will be tortoiseshell? Only 25%. Why is it impossible to have a tortoiseshell male offspring? It's because at the fur locus, or the fur locus only exists on the X chromosome. It is X-linked. And it is tortoiseshell fur is the result of the codominant relationship between black and orange alleles at the fur locus. Codominant. And so you have to have both a black and an orange allele to be tortoiseshell. And because these are linked to the X chromosomes, you'd have to have an X chromosome that carries the black allele and an X chromosome that carries the orange allele. But males only have 1 X chromosome. That's what makes them male, because one of their 2 chromosomes are the Y chromosome. And the color locus, the color gene, is not found on the Y chromosome. So a male can only have an XO or an XB. They can't have an XO and an XB because one of their alleles, or excuse me, one of their chromosomes is Y and is ineligible. Or I shouldn't say ineligible. It simply doesn't carry the fur color locus. So it's not possible to have a tortoiseshell male offspring because a male can only be XO or XB with a Y, because this Y is what makes them male. And so let's investigate this cross, just because we can. So if we, instead of having, actually, this is another thing. So if you are a female from this cross, let's get rid of this really fast. So if you are the offspring of this cross, which we see right here, if you're a male, what is the probability that you're black? Well, we can simply change our color first and then look at just the males. So if you're a male, what's the probability that you're black? Well, we see only one XB and one XO. So if you're a male, it's 50% probable that that you possess black fur, and 50% probable that you possess orange fur. Now, if you're a female, what is the probability that you have orange fur? There are two female possibilities. Orange fur is one of the two, and that's 50%. What is the probability of being a black female resulting from this cross? 0%, because only the mother can give a black allele because the father was orange. The father is orange, that means he has one orange allele on his X chromosome, and then he has a Y chromosome, which does not carry an allele for color, for fur color. So the male can only contribute, in this case, an orange chromosome. And because an orange chromosome mixed with a black chromosome creates the codominant tortoiseshell phenotype, you can never have an entirely black furred individual. So it is 0% possible or likely that one of the results from this cross will be a female that has black hair or black fur. But if you switch this and you, instead of having an orange male, had a black male, then this would be XB, and then this would be XB, and this would be XB. So then you would have 0% orange because you have XB, which is a black individual, and you have XBO, which is a tortoiseshell individual. Instead of being XOXO, you're XBXO, and that creates the codominant tortoiseshell phenotype. Cool. So I think I even had one more question. Yes, and this is blood type stuff. So a child who is blood type A has a mother who is blood type B. In a paternity suit, I wish I didn't have this cutting it off. Sorry, one second. Okay, a child who is blood type A has a mother who is blood type B. In a paternity suit, a man is accused of being the father. He has blood type AB. Is he the father? Give you a chance to work it out. You can pause the video and come back. So let's think about this. So a child has blood type A. I do not like that color. We're talking about blood here, so let's go red. Child has, actually, let's even describe that this we are talking about is the phenotypes. Child has A. The mother has B. And the prospective father has AB. So can this man who's being accused be the father? So in order for us to answer that question, let's look at the child. So the child, in order to have an A phenotype, an A blood type, their phenotype, or excuse me, their genotype could be that they have AA or AO. Obviously they'll have an A blood type if both of their alleles are AA. And they'll also express an A phenotype if their genotype is AO, because these codominate. So you'll have the A sugars being applied to your blood cells by this allele and no sugars applied by this allele. So you'll still express that A blood type, the A phenotype. So these are our possible genotypes of the child. So these are our genotypes. Here's the child. And the mother, we know that the mother has the B blood type. So the mother is kind of in the same boat. The mother could have BB, and that's what makes the mother have the B blood type. Or the mother could similarly have BO. So if the mother were to give, if the mother had this genotype, this BB, she could only give a capital B to her child. And having a single capital B, because we know that blood exhibits, maybe both of these questions shouldn't have been codominance, but anyway, if the mother were to contribute, obviously she did contribute to her child's genotype. So when the mother, if she had this genotype, contributed one of her alleles, the B allele, the child would have some kind of B in their blood type, because blood type is an example of codominance. So B would codominate with A, B would codominate with O. So there's no way that the child could have simply an A blood type if this were the mother's genotype, because the mother would give a B, and that would codominate with an A to express an AB blood type. But we know that the child simply has an A blood type, so it can't be BB, but it definitely can be BO. And this would indicate that the mother gave the O blood type, or the O allele, to her child. Because obviously if she passed on this B allele, the same thing would happen. This child would have at least an AB genotype. But we can see by the fact that this child has a simple A blood type that the mother gave her O allele to her child. And that also rules out the possibility of the child having an AA blood type. So this has to be the child's genotype, and this has to be the mother's genotype. So the child has to get AO. The child has to get AO in order to be this A individual. And if he gets the O from the mother, that means he gets the A from the father. But the father has blood type AB. We know this for a fact. And because blood type we think of as being just these two loci, we know that this is the extent of the father's blood type. So the father's blood type, or excuse me, genotype is AB. So we know the genotypes of each of the three individuals. But we have this child whose genotype is this. We have the mother whose genotype is this, and the purported father whose genotype is this. When they cross, we know that the mother gets or gives this O allele to the child, but in this cross, the father could provide the A allele that we see, or he could provide the B allele. And in that case, the child would be BO instead of AO. Because, the mother gives the O, the father would either give his A or his B allele. So it is entirely possible that this man who's being accused contributed his A allele to this child and is therefore the father, or it could be that the father possibly gave a B allele or gives a B allele in any of his offspring or to any of his offspring, and therefore he would not be the father because obviously a B allele can't make for an AO blood type. So it's possible that he is, possible that he isn't, and therefore we cannot rule him out. Like we said, it's possible, but then again, it's not possible. And I don't even know where this comes from. So it should be nice and clear cut. That's a pretty simple example. I wish I had another kind that wasn't just codominant. But then again, the other kinds of dominance are pretty understandable. What is this? Oh, that's not it. We have our codominance, which we explored a lot. We have our conventional or complete dominance, which is pretty well understood. And we have some incomplete dominance, which we can definitely do more examples of. But this video is running kind of long and I have class. So that's going to do it for now. This will be probably the last of my spring break review videos. Thank you for watching, if you did. And yes, we have a review soon and the exam on Thursday. Anyway, I'm going to stop babbling. Goodbye.